Good evening, everyone. Thank you to all our speakers for showing up and for giving their time to us. Our first speaker will be Professor Gus McCrowther, who used to work in Manchester but is now based in Singapore and very graciously got up in the middle of the night for us. But first, Matt Gagner will explain a few words about how to use Zoom and how everyone can ask questions. So over to you, Matt.
Good evening, everyone. Okay, good evening. For those that haven't used Zoom, we have an easy function to ask questions. You'll see there's a Q&A button. If you press that, you can type in a question, and the panel and I will try our best to answer them as we go along. We can potentially ask you to ask it directly, so you might like to talk, and I may well message you to ask if you'd be happy to do that. But on the whole, we'll try and address them as we go along, either through text or by directing it to the panel. You can also electronically put your hand up if you click on your name. And again, we'll look out for those hands, and we can always invite you to join the conversation if you wish. Without further ado, Professor McCrowther is going to speak.
Hello. The focus of this presentation is research in Dupuytren's contracture, Dupuytren's disease. And I think the first point to make is why we focus so much attention on this small disease that's only present in part of the planet and only inflicts a number of people and has no terrible consequences, although it's a huge functional problem for the individuals. Why is there so much research done on this? And the answer is that it's a wonderful model system for fibrosis, which is a component of so many diseases. Almost every organ in our body can have some kind of fibrotic process going on, lung fibrosis, cardiac fibrosis. But let's focus on Dupuytren's disease. You're going to hear one or two wonderful presentations. Some of the greatest minds, the younger generations than myself, are doing wonderful things on unraveling the intricacies of this disease. But I was going to focus just a little bit on 3 points which are clues to this disease, clues to why it happens. The first one is age, something getting very close to my heart. And it's quite clear that as you get older, you're more likely to get it. There are obviously genetic and other environmental factors that influence it, but age is unarguably very important. The older a population gets, the more common the disease is. And that's a great clue, because if we understand more about aging, then that's relevant. So I won't say more about that. The second thing I want to mention is the anatomy of Dupuytren's contracture. I think until a decade or two ago, it was thought to be a random disease that attacked various bits of your hand, but not in a terribly meaningful way. And in the 70s and 80s, there were a huge number of papers really describing the anatomy of Dupuytren's disease. And one or 2 points I want to make on that. I have here a textbook, which I will try and put in front of my camera, and we'll see whether we can see it. Here we are. And here we see a diagram of the fascial fibers underneath the palm of the hand. And basically, there are lots and lots of anatomically normal structures crisscrossing in the middle of the palm. And the disease doesn't go through the transverse fibers, it goes through the longitudinal ones. So if I show my own hand, I spent years practicing this. So hopefully you can see I've got very strong transverse fibers just across my web space here. Now, the disease runs longitudinally. It runs from the wrist up into the fingers. Anyone who's got it will realize that. And the transverse fibers are minimally or not involved at all. And so why does this happen? And that's an extremely important clue. And what it's telling us is that there is a mechanical influence on Dupuytren's disease that effectively spreads through the hand, spreading along the stressed fibers. So anything that's under a lot of stress is likely to contract. So the disease, the cellular changes in our normal palmar fascial structures, the normal structures that are involved by Dupuytren's, are responsive to stress. And a huge amount of work's been done on that in the last few years. So we have age, we have anatomy, that certain fibers are involved. It's not a random process. And then we have this response to stress or tension, which is now called by scientists mechanotransduction. I'm going to try and just share one or two slides to explain a little bit about mechanotransduction, and hopefully this will appear in just 1 second. I should be able to get into my slides here. It suddenly disappeared off screen. So I'll not worry too much about the slides just at the moment. But mechanotransduction is a process by which any tissues that are under stress the cells actually respond to the stress. We've known this for a long time because the operations for Dupuytren's disease have either been to actually cut it out or to relieve the stress on the tissue by cutting it. And this goes right back to Dupuytren himself. Baron Dupuytren, who described the disease so beautifully, used to just cut the palm of the hand across to snap all the fibers. And this removed the stress in them. And this was shown rather elegantly by some 19th century surgeons that what happened was the bits of tissue that were cut would then soften and undergo some kind of change. And this could be curative, not always. And so our treatments right up to the present time are very much designed along the lines of breaking up tension lines. There are still two schools of thought that go right back to 1826, when Dupuytren was first writing on this. There are two schools of thought as to whether you can get away with just dividing tissue, or if you actually need to take it out and cut it out. And surgery has gone from one extreme to the other, but it's been shown that there are many treatments which are based on relieving stress. I've found I think somebody's helped me here. I've found some of my stress slides now, and let me just come down here. Yes, here we see a Dupuytren cell. This is the state of the art in this whole thing. And the cell is sitting on tiny little pillars. If we look at the left, we have a nano patterned set of little pillars. And you can machine these nowadays with nanotechnology to the very finest size. So we can see a 5 micron, that's a tiny, tiny amount of tissue. There's a marker on the bottom of this slide here under the cell. And this thing that looks like a fried egg is a cell sitting on top of these nano pillars. And these pillars are sensitive to electrical currents. So we can now look at a single Dupuytren cell. And everyone with Dupuytren's contracture has literally millions of these in their tissues. And we can look at a single cell and we can see how it's moving and how it's contracting. And we can see that the cell is bending a lot of these pillars. That's all being measured by a computer. And you can actually measure how much force this cell is exerting. So at a research level, the tools are there to do incredibly detailed work on how a cell behaves mechanically.
Professor McGrather, can I just butt in briefly? I think most people can't see the cell. In fact, I can't see it either. We can just see a title. I don't know whether you need to advance. Whether you need to advance on one.
Shown up on my screen here, but for some reason it's not sharing. Well, you just have to imagine it. Think about a fried egg sitting on top of a bed of nails, and that's the way the cell is being interrogated. It's a pity. I'm not quite sure why that hasn't shared.
We can see the slide with the title, but nothing else. It's just blank otherwise. I wonder whether you just need to advance it. Perhaps it's animated.
Has that brought it up? No, it's showing on my screen. I'm now showing a picture of a patient. Is that showing?
No, it hasn't advanced from.
Oh, sorry. Well, that's a shame. My slides aren't showing up. Well, I'm almost finished my little contribution to this. Let me just explain it in words then. Understanding about the mechanics allows us to actually treat Dupuytren's disease in a rather different way. We can put a jig, a kind of a splint onto a patient's hand and actually straighten Dupuytren's tissue. Now, we've known for a long time that you really can't straighten a finger just with an external splint. It's not possible for patients to tolerate the discomfort of a splint trying to straighten the finger. But if you put little pins into the bone and then put a splint onto the pins, you can actually straighten a Dupuytren's contracture in 3 weeks. This has been shown using surgical implant systems. And what we've found is by growing the tissue in the lab, we've got an understanding of this. And what it seems is that if you put a little bit of force, a little bit of stretch onto a Dupuytren's contracture, you can't really change it at all. But if you put a lot of stress on and pull it really hard, you can actually model the tissue, because the cells, instead of making more and more scar, more and more tight stuff, they then produce an enzyme called collagenase, and the whole hand tissue can soften. So you just need to take my word for it if the slide's not coming up. That's a shame. So effectively, if we understand the mechanics of tissue strain and stress, we can apply this to treatments. And there are situations now where we sometimes straighten a joint out using an implant type system before we actually dissect out the Dupuytren's tissue. So I've said my bit, and I look forward very much to hearing my erudite colleagues on the high science they're all working on. So back to Matt or Anna.
Thank you very much for that. It was very informative. And I shall try and get my slide up. Right, so the next presentation hopefully is by Doctors Elizabeth Laird and Emma Beamish, the Liverpool group, the Dupuytren Research Group. And they're at the moment involved in cytokines research, which sounds very promising. Elizabeth and Emma, over to you.
Hello, thank you very much for inviting us to talk today. So I'm really delighted to be able to speak to you about our research that we're doing now. So hopefully, is that showing up on the screen? That should be our slides, yeah?
Yes, that's good. If you just click the presenter button, slideshow button, yeah, perfect.
Okay, so I'm Elizabeth and this is Emma. So I'm just going to give a quick introduction to collagen, which is the focus of our research group. And then Emma's going to talk about the project that she's working on at the moment. So we're really interested in our research group in the synthesis of type 1 collagen. So this is important in relation to Dupuytren's disease because the type 1 collagen primarily makes up the cord, which is one of the problematic factors in Dupuytren's disease. Apologies for the complex looking nature of this slide, but essentially type 1's collagen is made from 2 different genes that are in the DNA, so 2 genetic factors. These genes are copied into messenger RNA, which we can measure, and then they're copied then into protein. And normally type 1 collagen is made up of the products of both genes. It's made as a proform, which is then converted to a slightly smaller molecule, and then hundreds or thousands of these molecules come together to make a collagen fibril. And this is one fibril from a side on. So these are like small pieces of string, and then these associate together into bundles, which together can make up fibrous tissue. So this is really important normally because it makes up many of our tissues. So it makes up collagen's a major component of skin and of tendon and of bone and of the cornea in the eye. And so this process is critical normally, but when it goes wrong in fibrosis, as we just heard about, then it becomes problematic. So one thing that might go wrong is that the type 1 collagen becomes abnormal itself. So what can happen is that this second gene contributes less to the collagen that's produced, and then we get a collagen that's only made from the products of one gene. And this is a problematic type of collagen because it's resistant to degradation. So potentially this type of collagen could accumulate and exacerbate the fibrosis in fibrotic diseases and in Dupuytren's disease. So we've been working on Dupuytren's disease for a while now. We've been able to measure messenger RNA from patients' tissues and cells, and also the protein. So with the protein, we extract it from the tissue after we've labelled it and then separate it out, and then we can distinguish from these bands on the gel how much of each of the contributions of each of the genes is making to the protein that's produced. So our original study that we did was to try and identify if this abnormal type of collagen was present in Dupuytren's. So we did find that the products of both the collagen genes were increased in Dupuytren's disease, but the product of the first gene was relatively increased more. So for our controls, we used carpal ligament from carpal tunnel decompression surgery. And we use this because it's relatively normal and it cannot be obtained from hand surgery, and we can process the tissue within a similar amount of time that it takes to process the Dupuytren's tissue in the lab. And then we've also looked at the type of type 1 collagen that's produced in Dupuytren's disease, and we found that there was more protein that's coming from the first collagen gene than from the second collagen gene. And then we could convert this into a percentage of the type of collagen that's abnormal. And we found that in some patients it can be as high as 50% of the collagen is abnormal. But in the majority of patients, the amount is increased a little bit compared to what you would expect. So that set us into wondering what could be the causes of this abnormal collagen production, and could it be used to affect the progression of Dupuytren's disease. So I'll pass over now to my colleague, Dr. Emma Beamish, and she can explain the work that she's been doing.
Hi again, thank you very much for having us both. Hopefully I'll get through this as clearly as possible.
Oh, sorry, that's my slide. I forgot to mention this. So I just wanted to point out that a lot of patients very kindly donated their tissues to this study with the help of various NHS trusts. So we've obtained both Dupuytren's and carpal tissue, and the black lines on the left are the samples that were used for the first 2 slides. We've also done some proteomics work, which we're not able to talk about now, and the samples up to May 2019 are the ones that have contributed to Emma's project.
Okay. So as Liz has already mentioned, the abnormal type of collagen is formed and persists within Dupuytren's tissue. And this collagen is made by myofibroblast cells that are present within the nodule in patients' hands. And this is made in response to stimulation from cytokines. Now, cytokines are very small proteins that act as messengers between two cells. It's effectively like sending a text message from one cell to another. And this causes the cell to respond in some way. And in this case, in Dupuytren's, it is to produce this abnormal type of collagen. So we don't actually know exactly the mechanisms that underpin this in Dupuytren's disease, but the research out there at the minute suggests that cytokines associated with inflammation actually have a role in driving this abnormal collagen production within the tissue. So this has led us to hypothesize that specific cytokines may drive Dupuytren's contracture through the overproduction of the abnormal type 1 collagen. So we want to know is which cytokines are actually responsible for causing this abnormal collagen production. So we've identified a panel of 10 cytokines that are effectively our suspects. We think they may be involved, and they are out there in the literature as being implicated to be involved in the abnormal collagen production. So to test whether they are involved, we have obtained a specific inhibitor for each cytokine that we suspect. And those inhibitors actually block the function of those cytokines. They bind to the surface of them down here and stop them interacting with the cells. So we can then add these as treatments to our samples and see what effects they have on the collagen production within the actual patient tissue. So these inhibitors have been anonymized to me, so I'm blinded to them. This will stop me unintentionally biasing the data in any way. So they are just coded A through to K. And then we've employed this design table, which lets us test the 10 different inhibitors more simply than testing each one individually. And it will also show us those that have the strongest main effects on the collagen production as well. So within this table, A through to K are the 10 different inhibitors. And then the different runs, there are 12 different runs. And for each run, we are going to test different combinations of each of the inhibitors. This table has then been randomized, and I am currently working my way through the table. So each run, we collect a different sample from a different patient and bring it back into lab and do those tests on it. So to do this, I've been collecting tissue from patients undergoing Dupuytren's surgery to have the affected tissue removed. So instead of discarding it, we actually bring it back to our lab. I then dissect up the tissue into small tissue pieces, and then I also isolate the cells, the actual myofibroblast cells, from any remaining tissue. We then add the cytokine inhibitors to the tissue pieces and also 2D and 3D cell cultures. And then we look at the ratio of the newly synthesized collagen in response to those inhibitor treatments. So we are using this fairly new technique in our lab, and it involves making 3D and bioengineered synthetic mini tendons. So we actually take the cells that we isolate from a patient and put them into this dish within the lab. And then they, over a period of about 4 weeks, they self-aggregate inwards to form this mini tendon that you can see within the pins. And this is beneficial because this well simulates more closely the actual environment within the human body that these cells would experience. So they have all the 3D cell-to-cell contacts that they normally would, and they're also in retention. So it's a lot more representative of the environment that the cells are in normally. Okay, so the outcome that we're hoping to obtain from this research is that we may identify a new therapeutic target for Dupuytren's contracture, and that would potentially be via injection of either an inhibitor or a cytokine treatment into the hand. And of course, the dose and the frequency of these treatments would need to be determined by clinical trials, so this is still quite far away from being put into use. But if it is successful and it proves successful, it would enable us to target Dupuytren's disease a lot earlier, to reduce the abnormal collagen production that we see and attenuate the progression of the disease so that patients can maintain the digit flexion in their hands, reduce requirement of surgery, and improve the overall patient quality of life. So currently where we're at with this project is I have, as of yesterday, we are halfway through our tests. So we've done 48 out of the 96 treatment tests. Now there are 96 because we're doing 12 different combinations and we're repeating those 8 times just to make sure that the data that we obtain is powerful enough and representative enough of all the different combinations of the inhibitors that we're testing. So I don't have data for all of it yet because we're still actually processing these samples, so we'll get all the data once I've finished all 96 runs. But I have got some initial data from the very first run that we did just as a test. So in response to inhibitors A, B, D, E, and G, which we treated on the synthetic mini tendons, we found that the amount of type 1 collagen for the COL1A1 was significantly reduced in the Dupuytren's mini tendons, which for a first look looks really promising so far. But again, we have to do all the runs and test all the inhibitors and then unblind the data so that we actually know which inhibitors these are having this effect. Okay, so we would like to acknowledge the 4 hospital trusts that we're working with in the vicinity around here, at Halton, Aintree, Liverpool Royal, and at Wrightington. All the orthopedic surgeons at each of those sites who process these samples within the theatre for us. Our clinical teams who recruit the patients and consent them, and coordinate me collecting the samples from surgery, and then our funders who funded the research and made it possible, and the British Dupuytren-Society for letting us broadcast our research, and also the patients who consented to take part, and the general public who come to our events as well and participate and give us feedback. So thank you.
That was great, Elizabeth and Emma.
Thank you.
And we're very hopeful to hear what comes out of it. Oops, I think I've gone one step.
We're very keen to see the data too. I'm quite impatient now because we're a year in and we've got a year left.
Sorry, my screen sharing is sort of... Right, there we go. Yeah, we'll keep an eye on that research and inform everyone as and when we get the results from it. Our next speaker is Professor Nanchahal, who, well, we don't really need to introduce him because I think everybody has heard about the RATE trial and everyone is waiting anxiously for results and talking about, can we get it off license already? So, without anything more to say, over to Professor Nanchahal.
Sorry. So hopefully you can see my screen now. Yes.
Yep, it's all looking good.
Okay, cool. So what I'm going to do is take you through to how we got to the RID trial, so the target discovery phase, through to where we are now with RID. We've completed phase 2A and we're now into phase 2B. So as Gus mentioned, fibrosis can affect almost any tissue, and the culprit cell is the myofibroblast. So this produces the extra matrix, the collagen, etc., and also contracts it because it's got the same contractile elements as smooth muscle cells. Now, in a lot of fibrotic diseases, so for example, in the lung and the liver and so on, it's very difficult to access human tissue. So people have been using animal tissues, but that doesn't really reflect human disease. And this is one of the challenges in developing therapeutics for fibrosis. But Dupuytren's disease is potentially tractable because we've got access to human diseased and appropriate normal controlled tissue. And any novel therapeutics that we identify can then be tested using molecular markers as outcomes, and I'll show you an example of this. So when a patient undergoes surgery, this is a sort of tissue that's removed, and when you look inside it, a lot of it is collagen stained here in light pink, but you also see these dark patches, which is full of cells, and when you zoom in, and here they're stained for this alpha smooth muscle actin, which identifies the myofibroblast. It's just packed with those cells. And there are those same cells showing these contractile elements in green, and they actually touch each other and communicate with each other through these specialized junctions, so they can coordinate their activity and act like a syncytium, which is why these cells can actually start bending fingers down against people trying to straighten them. So it's quite a lot of force generation. So what we then did was we took these aggregates of cells and we separated them out into their component cells. And what we found was that whilst most of these cells were these myofibroblasts, there were quite a few inflammatory and immune cells. And when we left all of these cells in culture for 24 hours, they produce a whole array of these signaling molecules, cytokines, which Elizabeth and her colleagues have just discussed. So we tested most of these in turn, and I'm not going to take you through all the data, but I'm just going to highlight a couple. So TGF-beta, which has been highlighted time and time again in almost every fibrotic disorder, and then TNF. So if you take TGF-beta and you take fibroblasts from any tissue, so we took palmofibroblasts from Dupuytren's patients, we took palmofibroblasts from normal people, and we took non-palmofibroblasts from the same Dupuytren's patients, and you treat them with TGF, they all become more myofibroblasts-like and more contractile. So there's no difference between these cell types, but of course, as we know, palmofibroblasts are only in the palm, and only in Dupuytren's patients do they become myofibroblasts-like. And when you take myofibroblasts from Dupuytren's patients and treat them with an inhibitor of TGF, predictably you see a downregulation in that phenotype. So as I said, we tested most of those cytokines, and TNF was very interesting because only in palmofibroblasts from Dupuytren's patients at relatively low doses did we see those cells changing into myofibroblasts, whereas non-palmofibroblasts from the same patients or palmofibroblasts from normal patients didn't behave in the same way. And when we took myofibroblasts from those patients and treated them with anti-TNF, these are all the anti-TNF agents currently approved for other disorders such as rheumatoid arthritis, we saw a decrease in contractility. And what it seems to do is, so there are those cells again treated with a control antibody, but if you treat them with anti-TNF, they did seem to disassemble their alpha smooth muscle actin, so they're no longer as contractile. Dominic Furniss and his colleagues have done a lot of work on the genetics of Dupuytren's disease, and their work and that of others has highlighted the role of Wnt signaling in Dupuytren's disease. And we went on to show that the TNF, but only in patients with Dupuytren's disease and only in the palmofibroblasts from those patients, acts via the Wnt signaling pathway to upregulate these key genes that are involved in fibrosis. So these data form the basis of our trial. So we got funding from the Wellcome Trust and the Department of Health. I'd just like to digress for a moment. So why focus on something like TNF when in fact TGF may be a therapeutic target? So for a couple of reasons. So we studied all those cytokines from freshly disaggregated cells, and you can see that you get relatively low levels of TNF produced and about 300 picograms per mil of TGF. You keep those cells going for a couple of passages, the TNF falls to almost to zero, and that's because all those immune cells have disappeared. But the myofibroblasts produce TGF, so you get this autocrine secretion. And this may go some way towards explaining the failure of direct inhibition of TGF therapies in almost all forms of fibrosis so far. So what about our clinical trial? So this was in 2 parts, the phase 2A, which is a dose ranging, and I'll show you the data for those because they were published last year, and the phase 2B, which we're currently doing now. So in the phase 2A, what we did was we took patients with disease which was at a point where they would benefit from surgery, and we identified nodules, and we injected the nodule directly with different doses of anti-TNF. And what we found was that roughly, so this is lab work, that roughly about 0.3 to 0.5 milliliters of fluid remains within this nodule. And the dose cohorts we used were 15 milligrams of adalimumab at 0.3 mils and 35 and 0.7 mils. Now, when I was injecting 0.7 mils, I saw that some of this fluid was actually coming out into the subcutaneous tissue, so it was extravasating out of those nodules. And as it happened, the manufacturer of Humira, AbbVie, just brought out a new preparation of double the concentration, so 40 milligrams in 0.4 mils. So we amended the trial protocol and included an additional dose cohort. So these are the doses of adalimumab and the respective volumes, and we also had controls with the same volume of saline. And what we found was, compared to placebo, there was no change in the expression of alpha smooth muscle actin protein. So the patients had their injection, 2 weeks later they had their surgery, so we then had access to tissue. And this is what I mean about Dupuytrens being amenable to these sort of experimental medicine type trials, where you can take relatively small numbers of patients and still get a readout, because you're not relying on a clinical outcome, you're effectively relying on a biochemical outcome. But at the high dose, the 40 milligrams, but in this very concentrated formulation in 0.4 milliliters, we saw downregulation in the protein of alpha smooth muscle actin, and we also saw downregulation in pro-collagen 1. So we now had a potential dose that we could test. So we got some additional funding from the Wellcome Trust after we published our data last year to do the phase 2B. So the original target that was set by the statisticians was 138 patients. But in fact, we've had a tremendous response from patients in the UK. So we managed to over-recruit in the UK. And we were also able to open a third center in Groningen. So we closed to recruitment at all centers earlier this year at 181 participants randomized. So the trial structure is that the patients will be randomized. 50% will get the drug, 50% will get the placebo. So the person doing the outcomes assessments nor the patients know which one's which. They will look the same, the injection's the same. They will receive 4 injections at 3-month intervals over 12 months, and then be followed for a further 9 months thereafter. And we should have the final results towards the end of next year. So I'd like to finish by thanking the team. So whilst I've shown you some of these data, it's a huge effort from many members of the lab and also the clinical trials team. Important to include the collaborators, Dominic Davidson and Paul Verker. So Dominic is recruiting patients in Edinburgh, and Paul in Groningen, and the Oxford Clinical Trials Unit, and of course all of our funders. Thank you.
Thank you very much. That was a brilliant presentation, and hopefully we'll have some good results of that, Professor Nanchahal. We can't wait for the results. And I know a lot of patients were disappointed not to be included in the trial, because I think you could have doubled the numbers at least. I think Professor Nanchahal has his microphone off. So, last but not least, over to Professor Furniss now, who also works in Oxford and has done research on genome and genetic Oh. Okay, the screen suddenly changed for me. So Professor Furniss, one of our trustees, has done research on genetics of Dupuytrens. So over to you.
Thank you very much. And hopefully, can I just check the screen is sharing and you can hear me speaking? Can I get a thumbs
yes, it
up?
looks great, Dom.
Yeah. Looks all right. Great. Thank you. As Anna said, I've done a lot of work in my group and with various collaborators, including Paul Verker in Groningen, who's just been mentioned, who really is a fantastic collaborator, looking at the genetics of Dupuytren's disease. And I could speak for hours about all the different results we've had and what have you, but I thought just for 10 minutes, I would focus on one particular mutation or variant that we found that I think has potential to develop into a new drug, and we are starting the steps down that pathway. So Dupuytren's disease, as everyone knows, really it's quite a genetic disease. Probably about 80% of the predisposition to getting Dupuytren's is caused by genetic factors. But there are also non-genetic predisposing factors such as alcohol intake and smoking, BMI. So if you have a higher weight, if you're more obese, you're actually protected from Dupuytren's. It's the only disease really that I know where that's the case. But we're interested really in what those genetic factors are that predispose everyone to Dupuytren's disease, and really finding out what they tell us about the biology of the disease and whether we can alter that biology in order to create new treatments for our patients. And the way that we study these complex diseases where genes and environmental factors interact is called a genome-wide association study, or GWAS. And so we all differ from each other as human beings at about 10 million different points along our DNA code. And just for context, so that you all can think about how much that is that varies, we have about 3 billion base pairs of code that makes us all human. And so most of those variations are actually just single letters in the code that are different between two different people. So here you can see that most of the code is the same, but this person here has an A at this particular site, and this person here has a T at that particular site. And these variants are called single nucleotide polymorphisms, or SNPs. And so here, because we all inherit one set of our genes from our father and one from our mother, you could have someone who has an A from their father and an A from their mother, or you could have someone who has an A from one parent and a T from another parent, or you could have someone who inherits two Ts. So these are the possible different genotypes. So what we do is we take cases and controls and we compare the frequency of the variants all the way across the genome, across all 22 of our chromosomes. And we go into this study saying we actually don't have a clue what genes cause Dupuytren's disease. We don't go in with any hypothesis at all. We just say we're going to look and see which of these variants are more common in our Dupuytren's cases compared to our controls. And we test maybe 1 million of these different variants in a really big experiment. Now, when you test 1 million different variants, some of them will just randomly be more common in cases compared to controls. And so we need to have very strict statistical tests which tell us whether this variant truly is more common in our Dupuytren's cases, or whether that's just a statistical freak. And so we use a very low level of statistical significance. And in order to hit these targets, we need to have literally thousands and thousands of cases and controls. And as Jagdeep mentioned earlier, we've now done a couple of those studies and published them. And we've used, we have over 10,000 or so patients and many more thousands of controls as well. And these studies have found in total 26 different parts of the genome, different variants within the genome, which predispose people to getting Dupuytren's disease. And actually, we've done some work in collaboration with Jagdeep, my lab, and his lab, where we've looked at which of those different cells that Jagdeep was just talking about express some of the variants that we found are associated with Dupuytren's disease from the genetic studies. So we're integrating the cellular and the genetic studies. I'm just going to draw your attention on the right-hand side to this gene here, this variant here, in a gene called MMP14. And this seems to be mostly expressed in the myofibroblasts, so those cells that you've been hearing about already. So this is what the output of the genome-wide association study looks like. Here's the variant, which is right in the gene MMP14, and it actually causes a change in the protein sequence of that gene. And it's really common as well. So about just over a third of people in Western European populations carry one copy of this mutated gene, and about 7% of people carry 2 copies of this mutated gene. Thankfully, just over the road in the Kennedy Institute of Rheumatology in Oxford, which is where Jagdeep's lab is the world expert on this gene, a gentleman called Yoshi Ito. So I went and had a good chat with Yoshi when we found this result to say, okay, how are we going to take this further? So he taught me quite a bit about MMP14, which is also called MT1-MMP. And its main function in the body, it's probably the main gene in the body which deals with collagen degradation. And it's really important in a massive variety of different conditions. So for example, in cancer, when you have a primary cancer, in order for that cancer to spread around the body, it needs to degrade the collagen that's in the tissues in order to get out and into the bloodstream and go around the body. And that process is dependent on MMP14. In rheumatoid arthritis, there's tissue destruction of cartilage and bone and around the joints. And again, that process is very dependent on MMP14. It's also important in inflammation and in forming new blood vessels, which is important in various diseases such as diabetes. And also just normally maintaining a healthy, what we call extracellular matrix, so healthy connective tissue within the body. We tested both the normal MMP14 and the mutant form that we found associated with Dupuytren's in the lab. So what we did is we grew a collagen film, and we took some cells that don't usually express any MMP14, and we made those cells express either the normal type or the mutant type of the MMP14. And when we just grow the cells normally, you can see the collagen film stays completely intact, and so the light doesn't shine through it, so it looks dark. When we take the wild type, so the non-mutant MMP14, you can see that the cells then eat through the collagen in the lab, and we can shine the light through it now, and so there are lots of these holes in that collagen film. When we take the mutant form, we find that it's much more like the ones which don't express it at all. So the amount of collagen breakdown is now much, much less. And we can quantify that, and we can look at the ones that express half and half and find that they're sort of partway in between. And so this made us think that, you know, this kind of fits well with the Dupuytrens that we see in our patients, where there's an excess of this collagen laid down in that palmar fascia. So what happens in real patient cells? So these are just cells in the lab. What about real patient cells that we take from their hands at the time of surgery? So we have a big bank of these cells that we've collected over a period of years from many hundreds of patients. And so we could look into that bank and we could pull out patients whose genotype was either the wild type, so this is the normal MMP14, is GG, or were homozygous, so had 2 copies of the mutant form, 2 copies, 1 from each parent, or were halfway in between and had 1 copy from 1 parent which was wild type, or 1 copy from 1 parent which was the mutant form. And interestingly, what we found was that in either the ones who had both copies mutant or had only 1 copy mutant, they still had a big decrease in the amount of collagen that they could degrade. So we find not only in lab cells, also in cells derived directly from the Dupuytren's disease tissue, that collagen degradation is really inhibited. So what have we done with this data? Well, we're getting it ready for publication, but importantly, we've started collaborating with a pharmaceutical company now who has a panel of about 400,000 different medicinal compounds, and we're doing a collagen degradation screen to try to see if we can reverse this loss of activity. And there is a possibility, clearly we're a long way from this at the moment, but there's a possibility this might lead the way towards personalized medicine for patients, depending on what their genotype is. We may have particular medicines that we can give them that would reverse that collagen deposition and allow their collagen to be degraded again. I mentioned that MMP14 is important in cancer, and so we wondered, does this variant actually protect people from, if they get a cancer, from that cancer spreading? And we started to get some collaborations with various groups around the world who look at breast cancer and colon cancer and pancreatic cancer and various other cancers. And we're going to try and test that hypothesis in those patients. And we're doing more work to understand what the mechanism is. So this might lead us on to different molecules that could be amenable to having their function changed by various other drugs and things. So that's kind of where we're at the moment. I think the future is really bright for Dupuytren's research. We've heard several talks now this evening from people looking at this from completely different angles. And it's usually when you have many different research groups looking at a problem from lots of different perspectives that real leaps forward can be made. And it's a real pleasure actually in the Dupuytren's community to be able to collaborate with lots of different groups doing lots of different kinds of work. And hopefully this will lead to new treatments for all of our patients. So thank you very much. Back to Anna or Matt.
Thank you, Professor Furniss. I think we have some really bright minds here working on trying to get treatments and maybe even a cure for us. Matt, do we have any questions waiting? You're on mute, Matt.
Sorry, we just have the one that you posted to our group earlier. Would you like me to read it out? So it says, do the same cytokines that make us produce extra myofibroblasts also cause them to produce collagen and make the collagen start contracting? Or could it be different stages of the disease are steered by different cytokines? I wonder whether the Liverpool team might like to answer that, or perhaps Jagdeep, if he's still there.
We can both use it again.
Jagdeep?
Yeah, can you hear me?
Yes, yep, loud and clear.
So we found that the TNF converts the precursor cells into myofibroblasts, and in turn that upregulates the production of collagens and also the contractile elements that make up the myofibroblasts, that characterizes the myofibroblasts of the alpha smooth muscle actin. And when those cells, Dupuytren's patients are treated with anti-TNF, you see a downregulation in all of those characteristics, the contractility, the expression of collagen, and also the alpha smooth muscle actin. So both in the lab, and I've shown you also the clinical trial data from the phase 2A.
Great, excellent. We have one question. The cell phones in the hand stress the cells. Would anyone like to take that one? Dom?
Okay. I don't think there's any evidence that using cell phones or any kind of mobile device has any detrimental effect on Dupuytren's. I'm not sure that it's been properly studied, but I certainly don't know of any data that suggests that that's the case.
Thank you very much.
Great.
Anna, I don't think we have any more questions at the moment.
I've got a question from a member who couldn't be here.
Okay.
There's been some research that suggests that patients with Dupuytren's, where the contracture gets bad enough to actually need surgery, might be at more risk of cardiac disease or cancers. Do patients like that need extra screening for certain types of cancer, or is it a low risk?
So I should probably take that question. We've just had our paper accepted for publication. It hasn't come out yet, where we have demonstrated that patients with Dupuytren's disease have a slightly higher risk of certain types of cancer and of heart disease and of overall mortality, in fact. This seems to happen around about 10 or 12 years after the original diagnosis of Dupuytren's disease, for reasons that we're not 100% sure about, but there does seem to be this kind of delay for getting before this increase in those diseases kicks in. And we sort of see that as an opportunity for health promotion. And because many of the diseases that patients die from have other risk factors that are modifiable, for example, smoking or alcohol or high cholesterol, then I think it's important to address those lifestyle factors. In terms of screening for, let's say, certain types of cancer, I don't think the evidence is strong enough to make that a clinically useful thing to do, because the increased risk is sort of up to from 1 to 1.5, say. So you could call that a 50% increased risk, but if your baseline risk is already reasonably low, then a 50% risk of something that's reasonably low is still not a massive increase. So I certainly wouldn't panic about it, but I think it's a good thing to sort of use as a way of promoting a healthier lifestyle and making sure that all your other risk factors for those diseases are reduced as much as possible.
Fair enough.
Can I speak? I wonder, can you hear me? I'm not sure if I'm muted here or not.
No, we can hear you loud and clear.
Okay. Could I possibly ask my colleagues two very simple questions? Why does it get more common with age? And the second one is, why is there no Dupuytrens in Singapore?
Could I take the second one, Gus?
Yeah.
I think it's probably, so as I mentioned, Dupuytren's is a really genetic disease. It's about the same level of genetic predisposition as type 1 diabetes, and much more than things like heart disease and type 2 diabetes, much more. The frequency of the variants that we find within the genome-wide association studies are much higher in European populations than they are in non-European populations. And so I think that accounts, at least in part, for what we see in terms of the overall geographic distribution around the planet. Can I let someone else do the age one? Mainly because I don't really know the answer.
Everybody's very quiet on that one. But I mean, what do you think's happening? Is it an accumulation of genetic damage, or is it are there things happening to our bodies with aging that interact with all this in some way? Some failure of collagen homeostasis.
Should we try to answer that one then? Yeah, there's lots of things, I guess, that change with aging, so we don't probably know what the causes would be. So collagen definitely changes with aging, so it becomes more cross-linked, so the tissues become stiffer with aging, so that might be a factor. Something else, I guess, that goes wrong with aging is the immune system. So the immune system becomes dysregulated and doesn't work as well, so because there seems to be a lot of inflammatory cells involved in Dupuytren's disease, that could be a factor. So I'm just giving some potential answers to that question without actually knowing the answer. So if anyone else has any other comments, that would be good to hear.
Those are both useful comments.
Hannah, we've reached 9 o'clock. There are a few more questions coming through. Shall we answer a couple more?
Yeah.
Yep. Okay. So, does this research in any way link with Ledderhose disease? Jagdeep, perhaps, do you think that your anti-TNF could be a target, or could be a treatment for Ledderhose as well?
Possibly, I would. I don't think if it's found to be effective in the RID trial, then I think then someone might want to investigate anti-TNF for Ledderhose rather than go through the whole discovery cycle.
Okay, so yeah, but potentially. Do anti-inflammatory drugs, oral, topical, injected, offer any benefit to slowing down progress of Dupuytren's that we're aware of? Jagdeep, I don't know whether you want to comment on that as well. I'm not aware.
Yeah, I think, as Dominic has highlighted, that there's a huge genetic component to this. So Dupuytren's is actually pre-programmed into the palm of the susceptible individuals, and some of our unpublished work suggests that the epigenetic control is very, very important. So I don't think things like targeting the cytokines is going to lead to a cure. So I think the best result we can expect from anti-TNF would be control of progression. But if that were to be effective in any form, not just anti-TNF, but if any other local injection were to be effective, and if it were then to avoid the necessity for for example, surgical intervention, then essentially, you know, if the disease is kept in abeyance, then that's not such a bad outcome.
Okay, excellent, thank you. And then finally, is there any connection between Dupuytren's worsening and the heavy use of hands? So I suppose, Dom, you've essentially saying that 80% of the risk is really genetic, and that there are environmental factors, of which the use of hands could be part of it. Yeah,
so the literature on this is a little bit contradictory, actually. So there are some studies which show that Dupuytren's is more common in manual workers, particularly those who use vibrating tools. And this may relate to what Gus was talking about earlier with the stretching of the palmar fascia and the mechanotransduction of those signals into the cells. But there's also some decent epidemiological evidence that says people of higher social class, so in more managerial roles and more what you might call white-collar jobs, are more likely to get Dupuytren's compared to the people who are of lower social class and therefore in more manual work. And so when we look at our big data sets, we have some unpublished data which replicates this, and there was a big study which came out of Oxford in, I think, 2004, which showed exactly the same as well. So I think the literature is a little bit controversial. However, I think as a hand surgeon, and I'm sure Gus and Jagdeep will agree with me on this, we've all got patients who say, my hands were absolutely fine, and then I had this particular trauma, and now I've got the Dupuytren's. Now, whether that's they didn't notice it beforehand and then they start taking notice of their hand and discover that they got the Dupuytren's which they already had, or whether that's really a trauma to the hand that is setting off this cascade of myofibroblast proliferation and collagen being laid down and eventually contracture, I don't really know.
Can I just make a quick comment on that? Is my sound coming through?
Yes, yes.
Yes. It is interesting. I've had a few patients over the years where the Dupuytren starts in an area of scarring in the hand, like after a burn or a laceration. And it may well be that we know that the healing process from injury causes things to stick together. And I mentioned at the beginning this crisscrossing arrangement of ligaments under the skin of the hand. And it may be that if you switch on a certain level of mechanical stimulation, that may start things off. And so adhesions between the transverse and the longitudinal fibers seem to be a fairly early stage in Dupuytren's disease, where the freely gliding fibers under the palm of the hand get anchored somewhere, and something as simple. So it may be that many of these factors interact at the end of the day, that you need the genes, that something is happening with aging of collagen. It may be provoking factors like playing too much golf or something like that causes some inflammatory process, adhesions, mechanical stimulation. There are many different ways one can begin to weave the whole thing together. And you can weave it correctly, or you can be guessing. And this is why this high quality science is so important. And I have a tremendous admiration for my young colleagues, Dominic and Jagdeep and others, and the Liverpool group too. I listen to their work. It's fascinating to see that there's more knowledge coming through that's going to affect not only Dupuytren's, but a whole lot of other diseases too. So all we can do is encourage our patients to keep the driving force of getting this research work being done. It needs a lot of patient interaction for the grants to keep coming, the funding bodies, the governments, all have to be convinced that this is an area to keep investing in. So good luck to the future.
I think the BDS are really good at supporting us in that. So, yeah. Thanks, Anna.
So, Anna, we probably ought to draw it to a close there, do you think?
Matt, there have been a couple of questions just on radiotherapy. Shall I just say a couple of words about that?
Yeah, do if you like. I answered one. I think, Anna, were you considering another webinar on radiotherapy, I think?
Hopefully, either a webinar or a video recording by Richard Schaefer.
Yeah. But yeah, go for it, Tom.
So just briefly, what we and others have found with big data sets and also with smaller studies is that people with very early Dupuytren's disease with no contracture, around about 80% of those people never get a contracture and never need any kind of intervention at all. And therefore, using radiotherapy in those patients would often be a treatment that is not necessary. So we need to have a way of working out who from that 80% would potentially benefit because they're going to go on and get contracture. So when you look at studies and people present their results of radiotherapy in very early Dupuytren's disease, you always have to think, well, if they hadn't had any treatment, about 80% of them would have been fine anyway. And that's where I think Matt made a comment in the typed answers about the evidence base not being particularly good. And that's where we really would need trials of radiotherapy versus pretend radiotherapy, sham radiotherapy, to be sure that it's making a difference. And that's all I wanted to say just to frame that discussion.
Brilliant. Thank you very much. So, Anna, do you want to draw it to a close?
Yeah, I would like to thank everyone for participating and for giving their time to us and informing us of the research. It's been fascinating. I could listen to a lot more, but I think we've already gone over time, haven't we? So let's call it a halt. Anybody who's got any questions that have not been answered today, just send me a message or an email and I'll try and get them to the relevant speaker.